Test.
I'm going to go. So welcome everyone to Grand Rounds. I'm Patricia Aryan. I'm a professor in the Department of Psychiatry and Behavioral Sciences, and it's my pleasure to introduce Darshan Ramanathan, who is going to be talking today about oscillatory neural processes underlying skill learning in the healthy and injured brain. By way of introduction, Dr. Ramanathan received his undergraduate degree in biological sciences from Stanford in 2001 and completed his MD and PhD from UC San Diego in the Department of Neuroscience in 2009. He then pursued his residency training in the Department of Psychiatry at UC San Francisco from 2009 to 2013, followed by a 3-year fellowship training in the Department of Psychiatry and Neurology in the same university. He most recently has been conducting research in the Laboratory of Neuroengineering and Rehabilitation at UC San Francisco, and his primary research goals are to understand at a basic level how neural activity mediates learning and cognition by recording coordinated neural activity across microscales. So, for instance, at the level of a single neuron, mesoscales across cortical columns, and macroscales across whole brain regions. He hopes to use this knowledge as a foundation for developing new neural engineering methods of remediating cognitive and learning deficits in neuropsychiatric disorders. He has been, and it's very dark now in this room, he has been investigating, different scales of plasticity underlying skill learning since his work with Dr. Tiznowski, is that how? Tijinsky, thank you, in graduate school at San Diego. And he's been expanding his work to understand how these neural oscillations organize neural activity both within and across brain regions during skill learning and after motor injury. He's had 8 publications in some really exciting journals, Nature Neuroscience, PLOS Biology, and Journal of Neuroscience. And he's received many awards for his work, including a Burroughs Wellcome Career Award for medical scientists, the VA Career Development Award, and an NIMH Outstanding Resident Award. So great pleasure to introduce Dr. Ramanathan. Thank you.
Thanks, everyone, and thank you very much for inviting me to come up here and give a talk. It's quite a pleasure, and I'm very excited to be here. So what I'm going to be telling you about is, Pat introduced it very nicely, neural oscillations. And what I hope to convey to you is the importance of this in understanding psychiatric disorders and thinking of developing new treatments for psychiatric disorders. So if you think about treatments for psychiatric disorders, they're very coupled with the kinds of models we have, and there's a nice strong relationship. So early 20th century, development of psychotherapy, and the idea that our understanding and thinking about the mind relates to the treatment for psychotherapy. Late 20th century, with a combination of serendipitous discoveries, TB medication helps to make people happier, an antihistaminergic medication helps treat psychosis. And some Nobel Prize winning research led to this more molecular model for psychiatric disorders, and the treatments fit that. So what's our 21st century model for psychiatric or mental illness? And how do we think about that in terms of developing and tailoring new treatments? So we can fit in the older treatments within this overall model, right? The 21st century model for behavior and psychiatric disorders is that you have cellular, molecular changes, deficits, gene deficits that lead to cellular problems at the single neuron level, which leads to neural circuit problems, whole brain problems, and ultimately behavioral changes or abnormalities. And we can still fit in our old treatments in these, and notice that they're interconnected, right? So psychotherapy is targeting behavioral problems, but obviously those kinds of behavioral changes affects the brain, affects the circuit, affects the neurons. Likewise, psychopharmacologic approaches can affect cellular processes, which go the other way. But if you look at this map, you realize what's missing, right? We don't really have a knowledge that's deep enough at the circuit level to really develop targeted treatments at the circuit level, which can then spread in both directions. We're getting there. This is the promise of neuroscience, and we're approaching this. And so there's different approaches for how do we take our understanding at the circuit level and develop targeted treatment for that. So, obviously there's a lot of excitement about what we can call interventional psychiatry or neuromodulation in psychiatry. And the idea is exactly that. Can we intervene at the circuit level and affect brain processes, cellular processes, ultimately help with behavior? So DBS, TMS, TDCS, these are all models of how can we target the circuit and really develop new or better treatments for psychiatry. The thing is, as we know, psychiatry is very complicated. Behavior is complicated. Things that seem like they're theory-based don't always translate to helping with psychiatric disorders in the way we would want. And to me, what that tells me is not that, oh, TMS is never going to work, DBS is never going to work. What that tells me is things are complicated, and we just need a deeper understanding of how do circuits operate, how do we really know what particular circuit to target in a particular individual, and really we need better understanding of these processes and better models. So this is what I'm interested in working on. And there's many different ways to think about how do you develop good models of circuit functioning in normal, healthy populations or animals in my case, and then in dysfunction. And I'm very interested in neural oscillations or electrophysiology. So why am I interested in physiology? It's because you can measure the oscillations and oscillatory activity non-invasively and invasively. This means you can take physiologic measurements using EEG, which is outside of the brain, outside of the skull, in patients. You can also do, a more invasive procedure, which is called electrocorticography or ECOG, where you're putting grids on the surface of the brain. And then, of course, you can also record directly from particular brain regions by implanting electrodes in those regions. And I'm going to be talking mostly about what I'll call implant LFP or local field potentials. These are very localized oscillations right around the electrode where we've implanted. And what I'm excited about is this is a way that we can really bridge findings across animals and humans and study a level of circuit functioning which is most likely to contribute to potentially new targets for a treatment. And I'll use motor learning as kind of an example of how we've understood physiologic processes and oscillatory processes associated with these and developed targeted treatments towards these. So the other exciting thing to me about understanding oscillations and studying oscillations is you can really bridge multiple scales of nervous system functioning, which is very important in developing models, right? So oscillations, we know that oscillations organized single neuron spiking activity in different and interesting ways, and we can study this. And people study oscillations as a way of understanding interregional communication. And this can be at the mesoscopic level, so for example, at the cortical column level, as Pat was talking about, as well as between different brain regions. So, motor learning, skill learning, and during this talk, I'll tell you where I think the relevant psychiatric implications of what I've done is. So I'm going to be talking about, I'll divide this talk into 4 parts, 3 of which will be actual, organized, like part 1, part 2, part 3, and the 4th part is the psychiatric relevance, which will be intermingled in there. So the first part is, what are the oscillatory processes associated when we learn something in the very early stages? And I'll describe what I mean by these early stages. What are the oscillatory processes associated with more later stages of learning? And again, I'll talk about this. And can we use this knowledge to promote recovery of function after brain injury? So in this case, more of a neurologic model, but the same idea, the same basic approach. And as I said, part 4 will be kind of intermingled between all of this. So I'll let you kind of read this. So the question is, how do we learn new skills? So it doesn't work exactly this way. It doesn't work on the principles of electroshock therapy. But I like the idea that even Bill Watterson was thinking about psychiatric things when he was thinking about skill learning. So the flip side of how do we learn new skills is, how do we break bad habits? This is something, as in the mental health field, we think about this a lot. And I would say they're the flip side of the same coin. If we really understand how habits and well-learned motor skills are built, we might be able to develop new approaches for breaking these and changing habitual behaviors. So motor learning, there's a lot of different types of motor learning. There's a lot of different ways of thinking about motor or skill learning. I'm going to give you one model, and this helps to organize how I'm thinking about it, and we'll be talking about this during the talk. So the idea is, the first stage, the earliest part of motor learning, you can imagine, the first time you take a basketball and try to throw it into a hoop, or you're playing tennis and the first time somebody's like, serve it. And you're like, how do I even throw the ball up, coordinate everything, and hit the ball over the net? You're going to be horrible. Every single time you try to do this, it's going to be totally different because you haven't learned it yet. So this cognitive stage, the idea is you need a lot of attention. You're not very good. You're very variable, and you're very slow, and you make lots of errors. At some point in time, you've practiced this new skill enough that we kind of get to a mid stage of learning. We call it it can be called the associative stage. But the idea is you've learned the basic kinematics of it. So you know how to throw the ball up, coordinate your hand, and hit the ball over the net, but you're still not very good at it. You make a lot of errors, and you're a little bit faster, but you're just not that automated. And then we come to the Pete Sampras stage of serving, right? Where you basically, every single serve, the motion looks like every other surface motion. There's very little variability. You're very good. You're very accurate. And it almost becomes this habitual, non-conscious process. Non-conscious is maybe too strong, but let's just call it habitual. So first I'm going to talk about the transition from these earliest stages, from the stage 1 to stage 2. So how do we get to the point where when we first started practicing, at least that motor skill that we've practiced becomes somewhat consolidated? And a lot of people have thought, oh, this process involves sleep. So the first example, probably one of maybe not the first, but an early, very convincing example of this was from 2004. And they were using a motor skill sequence learning task in humans. This is well used in humans to study a particular type of motor skill learning, where you have to practice a sequence. It's almost like learning a piano. You have to practice a certain sequence of key presses, and you're very slow. And as you get more and more practice on it, you get faster and faster for a particular sequence. So what they really showed is if you separate this into a first block of training, a second block, and a third block, you see this jump, this significant improvement after sleep. suggesting that something has been consolidated and people are just better. We call this an offline improvement in motor sequence learning. And there's a bunch of different controls I'm not showing you here, but this isn't just because it's the third time that they're doing it. So any session that's separated by sleep seems to be associated with these offline improvements on this task. And this has now been replicated across a number of different labs, very robust finding. So what they found, and again, what's been replicated over and over again, is there's a particular stage in sleep that seems to help consolidate these motor skills. If you think about sleep, so, there's REM sleep, there's non-REM sleep. That's the first subdivision. And non-REM sleep is generally what we think of as slow wave sleep, because you see these slower oscillations which are associated with synchronous activity. And stage 2 versus stage 4 has to do with, in stage 2, there's a lot of sleep spindles. And by stage 3 and stage 4, you mostly see these big, slow oscillations and a lesser density of sleep spindles. So despite many, many, many different studies showing this is the case, people have also shown in humans that there's different changes that happen in brain activity. But we wanted to know, is there a cellular phenomena that's happening during non-REM sleep that helps to promote this motor memory consolidation? And we took our idea when we were conducting our first study from the hippocampal literature. So there's now a ton of literature that shows that after rodents run around in a maze, that's in the upper portion of this, right? You get this nice patterned activity representing that neuron and its representation of space in the maze. And so across a population of neurons, you can get a nice sequential pattern of activity. And this pattern of activity is actually replayed when the animal is sleeping. So we wanted to know, is there some similar kind of process that's happening to help consolidate memories after motor learning during sleep? So the way we conducted the study is we implanted electrodes into the motor cortex of rats, and we trained them on this skilled pellet reach task, the skilled motor task that's very commonly used to study motor learning in rats. And we wanted to basically compare what is the activity that looks like when they're first learning the task versus when they're sleeping. And then how does that relate to some change in the firing pattern when we can record these neurons after that sleep block? So here's the task that we use. Is this going to work? Okay. No. I think I have to make this a mouse again. Probably. Okay. So this is a very well-trained animal. And what I want you to see is the trajectory looks very similar and consistent over and over and over again. And this is exactly the task we use. So these are 3 different trials. I'm just showing you that, more or less, it's a well-trained animal. So the first thing we wanted to see is, can we, using our model, replicate the basic findings of sleep is important for behavioral motor consolidation in our animals? And what we found is, indeed, that the sleep period seems to be associated with some kind of offline improvement in the speed or execution of this task, which is actually, we weren't not sure what to expect, but this is very similar to what people find and document in humans. This is quantified across animals. So this is just showing one animal's data, and this is across animals, just looking at the speed of the execution of this task. And it's important in motor skill literature, there's a speed accuracy trade-off. So we can all do something very fast, but we're usually not very good at it. And if we slow it down, we can do it much better, but then we're very slow. And so we show that actually the speed improvement is not because they're just doing something worse. They're overall maintaining their accuracy on this. So the next question is, well, is there something about the single unit firing pattern that seems to be associated with these speed improvements? So this is showing one neuron's data across multiple trials, early and late in the first section, and then in green is after sleep. And what you can see is this neuron actually starts firing much faster after sleep. So something happened during sleep to really promote a shift in how these neural networks are firing. And again, if we quantify the neural speed, which is basically the shift in when these peak firings happen, you see a significantly faster firing patterns after sleep, and that's associated with an improvement in the motor speed. So these are, of course, related. We're recording in motor cortex, so we think that the neural firing is causing the movement. But we can't say for certain, but all we know is something happened during sleep such that the movements are faster and the neural networks are firing faster too. So one of the things that was noteworthy is, unlike the hippocampal literature, we didn't see nice sequential patterns of activity. In fact, most of our neurons during the task were firing in exactly the same time. This is a very fast task. These movements happen on the order of 500, 600 milliseconds, whereas in the hippocampal literature, they're talking about seconds of maze exploration. So rather than look for what they did, which is a sequential pattern, we said maybe what's really promoting these offline gains is synapse synchronous firing, and that's promoting plasticity during sleep. So we wanted to develop a way of detecting synchronous firing during sleep and seeing if that was associated with these plasticity. So I'm not going to get into the methods or the details, but we used a analytic method called principal components analysis, or PCA. And this is just a very good method at looking at synchronous activity across anything, and we looked at synchronous activity across unit firing. And you can look at the principal components of this particular animal. You can see the same thing that's happening at the single unit level is happening at this principal component level. So the entire synchronous pattern of activity across multiple units is just shifted after sleep. So now what I'm going to show you is the strength of synchronous activity, the strength of this principal components analysis during the sleep block before the animals have learned, which is right here, and the sleep block after the animals have learned the task. So this is just during sleep, and what you could see is the neurons that are involved in the task are synchronously firing much more after they've learned it during the sleep period. And this is just an example for 3 different time points. And you can see that these are the 3 most synchronous epochs before in the sleep period before they learned it versus the sleep period after they've trained on it. And you can see that the neurons are just firing much more synchronously after they've learned the task.
And these are the motor neurons.
These are the neurons in motor cortex. Exactly right. Yeah, so these are layer 5 pyramidal neurons in motor cortex. So we quantify this and we show there's a significant increase in this activation strength. And so we said, okay, great. So we have some kind of mechanism to show that these neurons are firing more synchronously. But what does this have to do with stage 2 NREM sleep and sleep spindles? So then we wanted to know, is there a relationship between sleep spindles and when these synchronous reactivations were happening? And so to look at that, I filtered the data in different frequencies, one associated with sleep spindles, and there's an important distinction. There's fast spindles and slow spindles, if you look in the sleep literature. And fast spindles are really associated with the consolidation of this task and occur over motor cortex, and slow spindles seem to happen in prefrontal cortex. Fast spindles are in the 13 to 16 hertz range we filtered at, slow spindles in the slower range. And so the take home is we found an association between sleep spindles and these reactivation events. We found an association between these reactivation synchronous events and the slow delta oscillations, and none with the slow spindles. So this was really the first way that we could link spindle oscillations to a cellular phenomena that really shows this is what spindles are doing. They're promoting synchronous activity in the neurons that are likely, this is the model, we don't know, but are likely driving some version of spike timing dependent plasticity because they're activating synchronously. Okay, so this is what we found. We're psychiatrists, mental health practitioners, psychologists, social workers. What relevance does motor skill learning have to psychiatry? Actually, a lot. So this is a group at Harvard that's been working on this for the last 10 years or more. part of the same Stickled group that showed the sleep-dependent motor skill processes in healthy people, and they started looking at schizophrenia. And what they found is there are reduced, and this is other groups have shown as well, but they found reduced spindle oscillations in patients with schizophrenia. And this reduction in spindle oscillations actually correlate with executive function deficits, and they're also associated with impairments in procedural or in this motor skill learning task. This you can see, control subjects have this nice offline improvement, and patients with schizophrenia don't. So one could imagine saying, how can we target spindle oscillations and improve them and enhance them in patients with schizophrenia? And could this really help to ameliorate cognitive deficits in these patients? Or we know a lot that cognitive training could potentially help with the cognitive deficits in schizophrenia, but maybe the cognitive training is limited in these patients because of these basic deficits in sleep-related plasticity mechanisms. So could you combine cognitive training with some kind of mechanism to enhance spindles or drive spindles? And could this be a new way of, now that we have a model, understanding how to improve treatments in patients? So just as a summary, so what I found is that NREM sleep promotes stabilization and increased speed in the firing of of the motor actions, as well as in the neural firing patterns, that it's associated with synchronous activation of the task-related neurons, and these reactivation events occur most during the fast spindles and slow oscillations. So there's actually different ways of targeting spindle oscillations, including some pharmacologic ones. But what I could also imagine is we have abilities and technologies to do closed loop stimulation. We can record neural activity, and anytime we see a spindle, we can actually stimulate and see if we can enhance activity, synchronous activity, during the spindle oscillations. And could we do that to basically enhance cognitive deficits in schizophrenia? So one of the things that I've been awarded to study is in a rodent animal model of schizophrenia, can we do this to ameliorate the cognitive deficits we see?
Do the meds that people take with schizophrenia have an impact on transmission oscillations?
So this is a great and actually understudied question. And even, for example, how do the meds that patients with schizophrenia take relate to both the healthy as well as sleep-related, the awake as well as sleep-related oscillations? And what I can say is it's complicated. And I don't know this literature well enough. All I know is when I was looking for it, I'm like, this is important and really should be modeled probably again in an animal model where you can really look at dose-dependent effects and specific pharmacologic effects of this. So now I'm going to talk about a different kind of problem. So how do we go from the early to the later, more automatic stage of motor learning? So why is this a difficult and more challenging problem? Is because we have to measure plasticity now, or some marker of plasticity or change across many days. So for those of you who are single unit physiologists, you'll understand why this is challenging. When we implant electrodes in the brain, neurons will just show up on an electrode and then they go away. And this is like this basic issue with stability. Sometimes you'll get a lot of stability over multiple days, sometimes you don't. And there's just always this challenge with how do you measure something that takes place over many days? So actually, I'll just show you this. I'll show you this. Okay, so this is just an example of what we mean when we go from a mid-stage to a later stage of motor learning in these animals. This is actually a video from my PhD when I was also working on that. Oh, that's okay. We can skip over that. But the bottom line is you can see that animals go through a phase where they know the task, they can get to the pellet, but they're really not good at it. So in my PhD, I was looking at more static measures of plasticity. We call this motor map plasticity. We can basically stimulate in the brain while the animals are anesthetized, and we can take two groups, one which didn't learn, one which did learn, and we can look at what is the overall change in the representation of motor body parts after they learned. and you get this nice expansion in the forelimb region, which is what they're training, at the expense of other regions. And what I found, so a lot of people have looked at this in a lot of different paradigms, but what I found is that when animals are training, it really requires cholinergic systems for this plasticity to happen. But as you can imagine, that's a very static measure of plasticity. What I would like is a more dynamic measure where we can really look at a trial by trial level and really understand what changes and when it's changing as the animals are learning this task over time. So we said, well, single units are hard. Maybe we'll find something at the oscillation level, which will really tell us about how the circuits are changing. So what we do is we put a grid of electrodes in motor cortex, same kinds of grids that we used in the earlier experiment, and same kind of task. But now, rather than just the overnight change, we're going to look at changes over many, many days and sessions of learning. So the first point is, when we think about oscillations, so oscillations are like if you think about little squiggles in the brain. So how do you study squiggles in the brain? And people said, well, it's nice to know what frequency are these squiggles happening at. So they can either be really fast or really slow. And you can use different analytic methods to decompose these squiggles into particular frequencies and look at the activity in these frequency bands. So when we looked at this, this is how we started. We said, oh, most of the activity is happening in these lower frequency bands. And this is even after normalizing to the fact that, of course, you always get higher power and frequencies, but even after normalizing for that, you still see that the change related to the task really occurs in these lower frequencies. So then I said, okay, let's just look at this low frequency band. Let's plot the activity across multiple trials, across multiple days. This is from the same electrode. So this plot has three interesting things I'm going to point out. So this line is time locking the activity to when the reach action starts. And what you can see, first of all, if you look at the black and the red dots, is this is a marker for how the animals are doing in terms of one aspect of learning, which is the variability of the kinematics, right? So sometimes the animals will get to the pellet and it'll take a long time to retract. And around here, so this is like the early learning stage. Trial to trial, there's a lot of kinematic variability. You can see at the late stages, the kinematics look exactly the same. It's like the animals reach out, grasp, retract, grasp, retract, grasp, retract, just like I showed you in that early video. What you can also see is the power increases over time and becomes really stable when these later stage kinematics have become very stable. So this is to show the power increases if you just compare the first 50 and the last 50 trials, and the oscillatory trace itself becomes very phase locked to these different parts of the movement. So again, this is early. This is multiple trials, the oscillation overlapping each other. And so this is a marker of plasticity that's occurred in that particular region of space so that the same exact oscillation happens trial after trial after trial. And this, we can measure this statistically using a method called phase locking, and you see very significant increases in phase locking as the animals have learned the task. So the first thing I want to know is, okay, I was just telling you about one region, but I've recorded across motor cortex. So how do we think about synchrony or synchronous oscillations that are occurring across motor cortex? So again, I'll use, without getting too much into the methods, I'll just say we used a principal components analysis approach to look at synchronous patterns of activity. And now we're going to look at different regions that show different patterns of synchrony. And is there any coordination in this process across different regions? And so this, in the early trials, if you look at the neural trajectories, which are on the right, you'll see trial to trial, they look kind of similar, but not really that similar. Once the animals have learned it really well, what you can see is at the neural level, you see exactly the same patterns of activity across motor cortex in this low frequency band. And the neural activity itself becomes highly stereotyped. And again, this is just showing the same exact thing in a more static way and highly significant. This degree of neural stereotypy across motor cortex. So one of the things I was telling you about before is we can look at oscillations and try to understand, well, how is it regulating spiking neurons and spiking activity? How is it organizing activity across close regions of motor cortex? We also implanted animals with electrodes in both motor cortex and dorsal striatum, which we know is interconnected with motor cortex and is strongly associated with habitual motor behaviors. And what we find is, so blue is motor cortex, red is striatum. And early on, these are two channels, you don't see much coordination. This is the average LFB activity across the entire session. And later on, you see that it becomes very stereotyped, and these two regions are basically oscillating very much in sync when the motor action is happening. So again, we think this is really a marker for well-learned habitual motor processes, is synchrony between these corticostriatal networks, and we can measure it in real time.
Is this the frequency that's similar, or the actual incidence of the fire?
So if you look, so this is averaged across trials, but you can literally look at it at a trial by trial level, and you'll see if you just filter the oscillation, that they'll be out of sync, and then as soon as the motor action happens, that oscillation gets perfectly in sync, and then it goes out of sync again. And right, yeah. And so this is a measure of that across on a trial by trial basis, looking at a measure called coherence. And so you can use coherence, phase locking, any way you look at it, these two regions become much more synchronized. I'm not going to get into this, but what's very interesting about this is it's very regionally specific. So different electrodes will show patterns where they become more coherent and others don't, suggesting that we can really now look at these very local processes of synchrony, microsynchrony. Okay, so again, I talked a lot about corticostriatal and intracortical plasticity using oscillations as a marker for motor skill learning. But what relevance does it have in psychiatry? Well, if you think about, so this is true in OCD as well as motor tics and autism. You get a lot of stereotyped motor behaviors, right? For OCD, it tends to be more complicated habit-like behaviors. In Tourette's, it's more motor, and in autism, it's maybe somewhere in between those. So with OCD, what's very interesting is when you measure in the same frequency, these low frequency oscillations, less than 5 hertz in this paper, connectivity of corticostriatal circuits, they're strongly associated with Y-box score for motor tics and obsessions compulsions. Now, ventral striatum DBS, which we know works for OCD, actually reduces this connectivity. And this could be a very strong model for who should get DBS and actually how it's working. You can imagine tailoring the DBS based on these low frequency connectivity between frontal striatal circuits to really say, are we doing it in the right place? Are we doing it with the right dose? Is this affecting the circuit we want? So this is where understanding these kinds of circuits at the oscillatory level can really improve how we develop interventions. So we talked about this, and we talked about this. And what's very interesting is now there's more and more literature that in autism, the stereotyped motor behaviors in autism are related to dysfunctions in actually ventral striatal medium spiny neurons. And it gets very interesting to think about DBS in autism, starting with animal models and really thinking about, is that a new approach to affect these kinds of circuits in the same kind of way? All right. Okay, so another thing that's dear and near to my heart, which I've mentioned a little bit, is so we have all of these oscillatory processes, we've studied this, but how can this really help us when we're trying to develop electroceutical intervention? So really modulating these circuits in an electrical or interventional way. So we were studying this in a stroke model. So I've been interested in injury and plasticity related to injury for a while, and in my PhD, what I found is, again, using a static measure of plasticity, you can stimulate and actually evoke very complex movements. This was shown in primates before, and then I found this is true in animals too. So you can see what looks like a reach, grasp, and retract movement. This is an anesthetized animal, just stimulating in the brain. And these kinds of complex movements really seem to, plasticity of these really seems to predict recovery of function. Do I have that? Oh, I don't have the correlation, but it predicts recovery of function after a focal motor injury in these animals. So, so taking the static model, now we want to say, well, what can we do in an oscillatory level to really modulate the circuit, similar to how we think about for DBS and Parkinson's or OCD. So first of all, we do a photothrombotic stroke, and we implant the electrodes in perilesional cortex, where a lot of other literature, including work that I did, shown plasticity is happening as the animals recover. And what you find is, immediately after the stroke, this is again the low frequency oscillations that we showed become highly stereotyped after the animals learn the skill, they're gone post-stroke. And really the recovery of these oscillations correlates incredibly well with the recovery of the motor function of the animal. So the left plot is the same thing you've shown across multiple trials and days, and the right is showing the accuracy at both a single trial level, which is the black and red lines, as well as a smoothed average. This is just to show, this is one animal, but across animals, the power significantly goes up as the animal is trained. Again, this is the last 50 trials compared to the first 50 trials post-stroke. And there's a strong correlation between, on a session-by-session level, the degree to which animals improve and the percentage increase in the power. So both of them are how much they improve and how much the power goes up. So we have this oscillatory marker that correlates with recovery. So can we develop an intervention that actually targets these oscillations and improves motor function? So one of the things we had found separate from this kind of work, this was another postdoc in the lab, had been doing direct current stimulation experiments in anesthetized animals. So there's lots of different ways you can stimulate the brain. This gets into the inherent complexity of interventions and how do we tailor and target interventions appropriately, right? This is why interventional psychiatry is very challenging. So direct current stimulation basically just applies a simple on-off pulse for a certain amount of time. And what this postdoc found, Tanuj found, is in anesthetized animals, applying direct current stimulation could increase power and spiking to these low frequency oscillations. So it just happened to be, he happened to be using a model of anesthesia, ketamine, that induces a lot of strong low frequency oscillations. So we don't think that DC current specifically increases low frequency oscillations. We think DC current just basically modulates whatever the dominant ongoing oscillation is, increases it, and increases the amount of spiking you get. But this is basically what he showed. So then we said, okay, we know that there's an ongoing oscillation. We want to modulate it. Can we apply DC current just when animals are reaching? So for a very short amount of time, and thus basically improve recovery. And we compared it to a sham where we stimulated right for a period of time before the trial happens, in case there's something alerting about being stimulated. And we also compared it to a high frequency alternating current stimulation. And so this is one animal's data showing that basically as you increase the amount of current, you get better accuracy. And in the same animal, what we found is this is kind of it's like an on-off switch. You don't stimulate the animals, so you can see kind of the baseline post-stroke accuracy. It's around 30% for this guy, 35%. And when you apply the stimulation at, for this animal, what we found was a good effective dose, 200 micrograms, you get the improvement. You apply it again, the improvement stays, you turn it off, it goes away. And, one of the things we were kind of wondering about is, could we induce some long-term plasticity? If the animals are training when they're better, maybe they would stay better. But we didn't actually find that to be the case. It really seems to be that this model is modulating the circuit and improving motor function, but as soon as the stimulation goes away, they go back down to normal. But we did find across animals that this method could really significantly improve their accuracy. And we compared it to these other controls, and we didn't really find that other ways of delivering stimulation had this kind of effect. So just to summarize here, these oscillations can be enhanced using this kind of direct current stimulation approach. This is an anesthesia. We can actually apply this in an on-demand manner to improve motor function after stroke. And now you can really kind of think about, okay, if we're trying to target oscillations more specifically, we could actually try to use a closed loop manipulation to stimulate at the particular phase of the oscillation that we think is important in driving behavior. So now we have a model to really understand how these motor circuits are integrated together and how to drive the circuit, which is a little bit more specific than just turning a current on and off. This is what we're currently working towards. Yeah.
Making sidewalks.
Yeah. Well, the ethical aspects of this is a whole nother set of literature, actually. That's interesting to think about and important to think about. So just to finish up, so, you know, it's my belief, and what drives me, what I'm excited about, is the idea that we can really develop new therapeutics in psychiatry that modulate circuits in a focused and targeted way. And my bias is using an oscillatory approach to develop targeted treatments for it. Though, as you can imagine, in the literature, there's all kinds of different ways of doing this. But ultimately, I think that's how we're going to get better treatments in psychiatry. So, for example, you could imagine a closed loop electrical stimulation, kind of like DBS, targeted to interrupt pathologic oscillations, which might be based on recordings from a corticostriatal circuit. So in DBS, does the stimulator really need to be on all the time? Or should it be only on when there's this pathologic oscillation? You might say, well, does it matter? Actually, they're doing the same thing in Parkinson's, where they have a beta coherence model between M1 and striatum, and they show improved benefits and improved outcomes when they have a closed loop stimulation approach rather than just an ongoing approach. I shouldn't say they necessarily do it. This is being tested in Parkinson's. This is a closed loop approach, but it certainly seems to work in animal models. You can also imagine a brain-computer interface approach to modulating oscillations that are dysfunctional in psychiatry. So schizophrenia is a great example. There's a ton of literature that shows task-evoked gamma oscillations are deficient in schizophrenia, and kind of resting gamma oscillations are higher. Well, we know very well, there's a good paper that was published a few years ago that shows that you can train animals to modulate gamma oscillations, and this drives spiking in predictable ways. So if there's a gamma deficit, maybe we could actually use a kind of neuroengineering approach to targeted neurofeedback to really modulate the exact oscillation we would want to target. And then you could imagine even more interesting things, which when I was talking to Dr. Fetz here, he's been working on, which is basically some kind of endogenous operant-based conditioning to allow for control of an implanted neurostim device. So can the animals train using a particular circuit to stimulate just all intrinsically? So that's the end of my talk. Thank you very much. I just want to acknowledge everybody in the labs I've worked with. So my postdoctoral fellowship work, Karnesh Ganguly, he's the PI of the lab. And Tanuj Gulati was a postdoc who really helped train me in many of the things that I ended up doing. And Ling and Stefan were grad students that I worked with on very closely on some of these projects. And then my PhD was done in the Laboratory of Neural Repair under Dr. Tishinsky. So thank you.
I'm sorry if I missed this, but when you were doing the training and doing the drug simulation, I wonder if trying multiple learning models may be necessary. Like it might work in extinction learning, but not in another task because the circuits are all a little bit different. And
Right.
Dependence on transition to dorsal laterals, right? Those things are all very nuanced, but just because it fails on one doesn't mean it won't work on another. It probably won't work on them all.
Right. I think that's a great point. And I actually think even in the animal, even in this particular model, we're just at the beginning of developing the kind of targeted approach and really testing it out to say, how is it really working? It must be inducing some form of plasticity, you would imagine. And so actually now we're implanting electrodes in M1 and DLS and trying to see, when we do the stimulation, what's actually happening in the circuit, and is there a way that we can really maybe potentially enhance plasticity in a kind of circuit-based way.
The other thing is, I don't remember all the dynamics of the generation of gamma, like Josh Gordon, our future leaders.
Yeah.
Work, but if you're not at the right level, I wonder how well it will train. So if you're more of a terminal part of a generation,
Right.
I don't know if that matters. I imagine you would think about
Right. Yeah. So Vikas Sohal, Vikas Sohal at UCSF has been, amongst other people, has shown that really gamma, you can drive gamma or block it using optogenetic manipulations of parvalbumin neurons. And it tends to be a very local microcircuit operation. It's not something that's driven by long-term connectivity. So one of the things, you definitely imagine with something, with a model like that, is exactly the key question is, if you're just stimulating the gamma frequency, what is that really doing to the circuit? And he's really showing that you at least optogenetically, stimulating parvalbumin neurons drives gamma oscillations, but stimulating pyramidal neurons doesn't to nearly the same extent. So would a stimulation approach work? It's not clear, but an operant conditioning approach where you train animals to modulate their own gamma, at least in animals, seems to show a similar kind of thing as if you were optogenetically driving the parvalbumin neurons. So this is where you can almost imagine some things we can exert on the system, some things maybe need to be cell type specific in an optogenic way, and some things maybe we can rely on the intrinsic plasticity of the brain to do it for you. Like, I just care about gamma oscillations. Can your brain do it? And empiric questions that I think are very interesting.
There's some evidence that cognitions and words and other things are sometimes linked with motor behavior because you learn words by saying them, and you need to have a connection with muscles or whatever. With you're looking at these oscillations, do you see any possibility of looking at correlations with learning or use of words or combinations of words?
Yeah, so there's definitely a few things that your question makes me think about. So one is this now very well studied idea of corollary discharge, which is the idea that when you're intrinsically generating movements, you're actually sending signals to sensory cortical processes associated with those movements to tell yourself, oh, these are intrinsically generated. And words are very similar in that sense, that when you're speaking, you're sending a lot of signals to auditory cortex to say, I'm speaking, so, modulate the circuit in a particular way. So you could definitely imagine, depending on what you're saying, and people have looked at the various aspects of these, the idea that this connection between motor and sensory cortical areas and the corollary discharge might be deficient in psychiatric disorders like schizophrenia, and that potentially knowing about this and trying to modulate it or train it could be very important for the kinds of learning you're talking about. I think the other thing is also interesting, which is in rehabilitation, it's known that there are certain kinds of tones or music that can help rehabilitation happen in a better way post-stroke. This is in patients and kind of empiric. And I don't know the literature well enough to know if there's a good model for this, but you can imagine based on this that maybe you can do an auditory tone that's really targeted to the frequency that I think might be important. And could you drive motor learning and plasticity in a particular way by driving just synchronous oscillations, which we know that certain auditory tones can do. So, so I think the idea of expanding this, even for this particular area, away from just motor and really thinking about whole brain processes is definitely the next step, the future of neuroscience, not just 1 or 2 regions, but 5 regions, 10 regions, and really understanding these at multiple scales. Yeah.
Similar to that question there any evidence that, like in the sleep consolidation paradigm, that training animals to do other things or just stimulating them in other ways with novelty or motor tasks also changes the way they learn on specific tasks?
I'm going to answer your question and tell me if I answered it. So what actually is known, so there's two aspects of this. So one is that reward plays a key role in the reactivation during sleep. This has been shown in hippocampal as well as in non-hippocampal tasks. So for example, if you completely, like one great study basically stimulated the dopaminergic system while recording from hippocampal place cells and basically tried to drive this connection of dopamine with the place cells when the animal was sleeping, not when the animal was awake. And they found that, oh yeah, after the animal woke up, they just went to that place much more than any other place. So this idea that arousal or reward or other things that are happening during the task are probably also being replayed or reactivated during sleep that helps to drive plasticity, I think is very relevant. And you can imagine things while you're learning to drive dopaminergic or other neuromodulators associated with plasticity could well play a key role in the sleep-related consolidation that we find in plasticity in general. The other thing that's also known, however, and this is the other thing that I was thinking about, is sleep actually helps to prevent interference. So if you just try to train on multiple things that are probably overlapping at a circuit level. So if you could train yourself to do like a motor task and then maybe a totally other hippocampal task, I don't know if you're going to get interference, but if you're training something which really involves a motor circuit, like two different types of motor sequence learning tasks, you're going to get more interference, and actually you'll learn both of them worse unless you separate it with sleep. Sleep can consolidate the first one, then you can learn the second one a little bit better. Did that answer your question? Or were you thinking of something else?
I think that answers it. I'm thinking clinically for people who can't sleep and we're trying to think, and they're always depressed and they can't change their life to hold themselves together. What can we do? Like how do interventions function?
Right. So maybe even though they can't sleep, can you ameliorate that deficit some other way? Maybe. I think it's a good question. Maybe what I would say is, and this is somewhat borne out with the clinical literature, that for depressed patients that really can't sleep, even if you don't treat the depression, treating the sleep can actually help a lot. So this is where I think psychiatry gets complicated, because you can treat the depression and the sleep gets better. In probably some patients, but not all of them, you can treat the sleep and the depression gets better. And why is that? And understanding that, I think, would be important to know more about. Well, what if you just can't get somebody to sleep? What else could you do to restore it? I think, yeah, it's a good question.
Just thinking of the global pharmacology literature, is there any way to enhance gamma oscillations with just medications?
Pharmacologically? Right, yeah, this gets to a great point, which is if you have a oscillatory framework, it's actually a biomarker that you could use to test out. So I'm glad you brought that up. So the ideal one would, of course, be one that specifically targets parvalbumin neurons, which doesn't exist, to my knowledge. I can imagine pharmaceutical companies must be trying to develop something because it just seems so obvious. I actually would be curious about ketamine and other NMDA agonists because of the strong connection with, at least, even though NMDA receptors are everywhere, they're certainly modulating parvalbumin function. And so I guess, let me make this short. I don't know the answer to that question. I would wonder whether, like, even low dose ketamine, if people have looked at the role of that with gamma, but I'm sure other people know the answer to this. I don't.
Affecting oscillations without actually cooling, like Michael P. might do in the song? Or
Right.
can you sort of separate oscillations from just the amount of firing that's occurring?
Right. So in other words, we look at activity and changes in activity, and then changes in, maybe I'll call it synchrony. And the idea of oscillations is they're really trying to synchronize the network as opposed to just driving activity. So the short answer is also, we've been thinking about this a lot, particularly in our model. And I think what that really requires is a better understanding of what's driving the oscillations in the first place. And at least for these low frequency oscillations that we're looking at during the task, we're putting together models and developing ideas to test, but we haven't come to a great idea yet. So probably what I'd say in our particular task is, one model is that actually the low frequency oscillations might be driven by, for example, thalamic inputs to M1 versus longer range cortical inputs like premotor or prefrontal cortex versus layer 2-3. So an ideal experiment might be, can you block just the inputs? And we'll still be asking ourselves, what does it do to the oscillations? What does it do to the firing rate? It's very possible that one kind of intervention will specifically get rid of the synchrony and at least it perturb the oscillations without affecting the firing rate. But that's going to be kind of the empiric question we're trying to do and look at. And yeah. Great, thanks. Thanks a lot.
Quick thinking on that. You couldn't play the first video with a laser pointer.
Oh yeah.
I was just like,
Yeah.
nice move. I'd never seen that before. Most people don't have videos,
Right.
so I never had anybody try to flip a video with a laser pointer. So. Learn something new
The
every
laser
day.
pointer probably had to need in retrospect.
Yeah,
It's kind of a nice feature.
It's kind of cool.